There was a soil bacteria so powerful that gardens inoculated once never needed fertilizer again. Plants grew 3 times larger than chemically fertilized crops. Yields doubled or tripled. Soil improved every year instead of depleting. Farmers applied this bacteria at planting and watched crops thrive on completely unfertilized ground for decades. One application transformed dead soil into living, fertile earth. Gardens that had been abandoned as useless suddenly produced abundant harvests. Bombed land declared unsuitable for agriculture was growing food within months. The bacteria didn't just feed plants, it rebuilt entire soil ecosystems from nothing. This bacteria wasn't genetically engineered. It existed naturally in healthy soil worldwide. Scientists had isolated it, cultured it, and learned to produce it in quantities that could inoculate entire farms. The technology was simple, proven, and available to anyone. And within 15 years of war's end, this bacteria disappeared from agricultural markets completely. Not because it stopped working, not because safety issues emerged, because it threatened a fertilizer industry worth hundreds of billions of dollars, because you cannot sell chemicals to farmers whose soil feeds itself, because you cannot build empires on bacteria that eliminate dependency forever. Number 1, the Azotobacter discovery in war-torn Soviet fields. In 1943, Soviet agricultural scientists faced catastrophic problems. German invasion had destroyed collective farms across vast territories. Chemical fertilizer production had been diverted to explosives manufacturing. Millions needed feeding, but no fertilizers were available for damaged, depleted soil. Dr. Sergei Winogradsky's research team at the Institute of Microbiology remembered pre-war studies on nitrogen-fixing bacteria. Azotobacter species, naturally occurring soil bacteria that captured atmospheric nitrogen and converted it to plant-usable forms. These organisms could provide the nitrogen that chemical fertilizers normally supplied. The team collected soil samples from the few remaining productive fields. They isolated Azotobacter strains and cultured them in laboratory fermentation tanks. Within months, they were producing bacterial inoculant in quantities sufficient for field testing. Initial trials were desperate experiments. Fields that had produced nothing for years were inoculated with Azotobacter culture. Seeds were planted with no fertilizer application. Researchers expected modest improvement at best. The results shocked everyone. Inoculated fields produced crops comparable to pre-war fertilized yields. Plants were healthy and vigorous. Growth rates matched or exceeded what chemical fertilizers had previously achieved. The bacteria were working exactly as theory predicted. But the truly remarkable discovery came the second year. Fields inoculated once the previous year produced even better crops without reinoculation. The bacteria had established permanent populations. They were reproducing naturally in the soil, continuing to fix nitrogen season after season. By the third year, yields in bacteria-inoculated fields exceeded anything achieved with chemical fertilizers. The soil was improving, organic matter was increasing, the entire soil ecosystem was becoming more alive and productive. Soviet agricultural stations began producing Azotobacter inoculant on industrial scale, distributed it to collective farms nationwide. Crop production recovered despite complete absence of chemical fertilizers. The bacteria had solved the fertilizer crisis completely. Number 2, the nitrogen fixation that replaced chemical factories. When Western agricultural scientists studied Azotobacter after the war, they discovered nitrogen fixation rates that made chemical fertilizer production obsolete. One bacteria produced what factories synthesized at enormous energy cost. Azotobacter fixed atmospheric nitrogen at rates of 20 to 40 milligrams per gram of glucose consumed. This meant the bacteria could produce substantial nitrogen fertilizer from simple sugar sources, crop residues, compost, even waste materials. 1 acre of soil properly inoculated with Azotobacter fixed 50 to 150 pounds of nitrogen annually. This matched typical chemical fertilizer application rates, but the bacterial nitrogen was free, captured from air rather than produced in factories. The energy comparison was devastating to the fertilizer industry. Chemical nitrogen fixation through Haber-Bosch process required temperatures of 400 to 600 degrees Celsius and pressures of 200 to 300 atmospheres. Enormous energy consumption. Azotobacter fixed nitrogen at ambient temperature and pressure. The bacteria accomplished at 20 degrees Celsius what factories required 500 degrees to achieve. The energy savings were astronomical. Bacterial fixation used essentially zero fossil fuel energy. The nitrogen form differed beneficially. Chemical fertilizers provided nitrogen as nitrate or ammonia, forms that leached from soil rapidly, required frequent reapplication. Much was lost to groundwater pollution. Azotobacter released nitrogen gradually as organic compounds. These remained in soil longer, were absorbed by plants more efficiently. Nothing was wasted through leaching. The biological nitrogen was superior to chemical nitrogen in every measure. Cost analysis showed bacterial nitrogen was essentially free after initial inoculation. Chemical nitrogen cost significant money every application. Over decades, the savings were enormous. one inoculation versus perpetual fertilizer purchases. But this threatened the fertilizer industry fundamentally. If bacterial nitrogen fixation became standard practice, chemical nitrogen sales would collapse. The industry couldn't allow this knowledge to spread. Number 3, the soil building from bacterial colonies. Azotobacter didn't just fix nitrogen. The bacterial colonies built soil structure and fertility in ways chemical fertilizers could never match. This complete soil improvement threatened the entire agricultural input industry. The bacteria produced extracellular polysaccharides, sticky compounds that glued soil particles together. These created stable aggregates, the crumbly structure that characterizes healthy, fertile soil. Chemical fertilizers provided no such benefit. Aggregate formation improved water infiltration and retention. Soil that had been compacted and impermeable became porous and absorbent. Gardens that flooded or dried out rapidly developed perfect water management, all from bacterial activity. Organic matter increased continuously. Azotobacter cells themselves contained carbon. When cells died, their bodies enriched soil with organic matter. Over years, this accumulated significantly. Soil became darker and richer. The bacteria attracted earthworms. Worm populations in inoculated soil increased 5 to 10-fold. The worms further improved soil structure and fertility. A positive feedback cycle developed. Bacteria attracted worms. Worms improved conditions for bacteria. Beneficial fungi colonized more readily in azotobacter-rich soil. The bacteria modified soil chemistry in ways that supported mycorrhizal fungi. These fungi formed partnerships with plant roots, further enhancing nutrient uptake. Soil pH stabilized naturally. Azotobacter activity buffered pH swings. Soil that had been too acidic or alkaline moved toward neutral optimal range. No lime or sulfur applications needed. Disease suppression resulted from bacterial diversity. Azotobacter weren't alone. Their presence encouraged entire beneficial microbial communities. These organisms outcompeted plant pathogens. Disease pressure decreased without any fungicides. This comprehensive soil improvement made chemical amendments obsolete. Bacterial inoculation provided everything: fertility, structure, water management, disease suppression. One treatment replaced dozens of agricultural products. The agricultural input industry couldn't tolerate this. Their business model required selling multiple products continuously. Bacteria that solved everything with one application threatened their entire revenue stream. Number 4, the crop yield increases without chemicals. Field trials comparing azotobacter inoculated versus chemically fertilized crops showed bacterial inoculation producing superior yields with zero chemical inputs. This data threatened to revolutionize agriculture away from industrial dependence. Soviet research stations conducted multi-year trials across diverse climates and soil types. Inoculated plots received only bacterial culture at planting. Chemical plots received standard fertilizer applications. Results were documented meticulously. First-year yields were comparable. Inoculated crops matched fertilized crops. This alone was remarkable. Free bacterial inoculation equaled expensive chemical fertilization. But the real surprise came in subsequent years. Second-year inoculated yields exceeded first year by 20 to 30%. The bacterial populations had established. Nitrogen fixation was higher. Soil improvement was becoming evident. Chemical plots showed no such increase. By the third year, inoculated yields were 40 to 60% higher than chemically fertilized plots. The gap continued widening. After 5 years, inoculated plots outproduced chemical plots by factors of 2 to 3. The yield advantage compounded over time. Each year, bacterial populations strengthened. Soil structure improved further. Organic matter accumulated. The biological system was building momentum. Long-term studies spanning decades showed inoculated soil becoming progressively more productive. 20-year comparisons revealed triple yields from bacterial inoculation versus chemical fertilization. The difference was staggering. Quality improved equally. Vegetables from inoculated soil contained higher nutrient levels, better flavor, superior storage quality. The biological approach produced better food, not just more food. Economic analysis was devastating to chemical agriculture. Inoculation cost perhaps $5 per acre once. Chemical fertilization cost hundreds annually. Over 20 years, bacterial approach saved thousands per acre while producing superior results. But this data threatened the fertilizer industry's entire business model. If bacterial inoculation was superior and essentially free, why would farmers buy chemicals? The research had to be suppressed. Number 5, the commercial production that disappeared. In the early 1950s, several companies began producing and selling azotobacter inoculant commercially. For a brief period, farmers could purchase bacterial cultures to replace chemical fertilizers. Then the products vanished from markets completely. The production process was straightforward. Culture azotobacter in fermentation tanks, containing molasses or other carbon sources. Harvest bacterial cells. Mix with peat or other carrier material. Package for sale. The technology was simple and proven. Multiple companies offered Azotobacter products. Prices were modest, $10 to $20 per acre of coverage. One application provided benefits lasting years. The value proposition was extraordinary compared to chemical fertilizers. Early adopters reported excellent results. Yields increased, fertilizer costs disappeared, soil health improved visibly, word spread through farming communities. Demand for bacterial inoculant grew rapidly. Then the products disappeared. Companies stopped producing them, existing stock sold out and wasn't replaced. Within 2 years, Azotobacter inoculant was unavailable commercially in Western countries. The official explanations were vague. Market conditions, production difficulties, quality control issues. None of it rang true. The technology was proven. The products worked. Demand existed. The reality was industry pressure. Chemical fertilizer companies recognized bacterial inoculants as existential threat. They couldn't compete on merit. Bacteria worked better and cost less, so they competed through suppression. Lobbying convinced agricultural departments to create regulatory barriers, new testing requirements, certification demands, standards designed to be expensive and difficult to meet. Small inoculant producers couldn't afford compliance. Larger companies were acquired or pressured. Chemical companies bought successful inoculant producers and shut them down. Independent companies faced supply chain disruptions. Banks mysteriously withdrew credit. The coordinated suppression was systematic. Within 5 years, commercial Azotobacter inoculant had been eliminated from Western markets. The product that could have revolutionized agriculture was made unavailable. Number 6, the research funding that stopped abruptly. University research on Azotobacter and nitrogen-fixing bacteria was extensive in the 1940s and 50s. Then funding dried up completely despite promising results. The research cessation was deliberate suppression. Multiple universities had active programs studying biological nitrogen fixation. They were documenting mechanisms, optimizing strains, and developing application methods. The research was producing valuable practical results. Papers were being published regularly in agricultural journals. The findings were solid science, peer-reviewed research confirming that bacterial inoculation could replace chemical fertilizers in most applications. Then, research grants stopped being renewed. Programs that had been funded for years suddenly lost support. Principal investigators were told to pursue other topics. The bacterial research ended abruptly. The funding decisions didn't make scientific sense. The research was productive and important. It addressed a major agricultural challenge. It should have received increased funding, not elimination. But the decisions made economic sense. Research funding came largely from fertilizer industry sources. Companies funding agricultural programs at universities. Their money came with implicit expectations about research directions. Studies proving bacterial inoculants superior to chemical fertilizers threatened the sponsors. The companies couldn't allow universities they funded to produce research destroying their business model. Department heads received pressure. Continue the bacterial research, lose industry funding, shift to other topics, maintain financial support. The choice was clear. Bacterial research was abandoned. Researchers who persisted found career obstacles, tenure denials, grant rejections, professional isolation. The message was clear. Research threatening industry interests would not be tolerated. Some scientists moved to other countries where funding continued. Soviet agricultural research on nitrogen-fixing bacteria continued through the 1980s. But Western research stopped completely. The knowledge existed. The science was sound. But the economic system suppressed it deliberately to protect fertilizer industry profits. Number 7. The farmer success stories that were silenced. Individual farmers who discovered and used Azotobacter inoculant achieved remarkable success. Their stories should have spread widely. Instead, they were systematically silenced and marginalized. Farmers who obtained bacterial cultures before products disappeared reported transformative results. One documented case in Iowa showed corn yields increasing from 60 bushels per acre with chemical fertilizer to 120 bushels with bacterial inoculation. The farmer stopped buying fertilizer completely. Soil health improved dramatically. Earthworm populations exploded. The land became progressively more productive each year. After a decade, yields had tripled, while input costs approached zero. He tried sharing his success, told neighbors, contacted agricultural extension, offered to help others obtain bacterial cultures. The response was hostile. Extension agents dismissed his claims as impossible, accused him of exaggerating, suggested his success resulted from other factors. Never investigated, despite documented evidence, neighbors were skeptical. The agricultural establishment had convinced farmers that chemical inputs were necessary. Success without chemicals didn't fit the accepted paradigm. Many refused to try even when offered free bacterial cultures. Agricultural magazines wouldn't publish his story. Editors claimed lack of interest, scientific verification required. The story didn't fit their editorial focus. The real reason was advertising pressure. Fertilizer companies wouldn't approve stories threatening their sales. When he persisted in promoting bacterial inoculation, he faced retaliation. Bank mysteriously increased interest rates on farm loans. Insurance costs rose. Equipment dealers gave him poor service. The informal economic pressure was unmistakable. Other farmers with similar success learned to stay quiet, share knowledge privately with trusted friends only. Public promotion invited economic consequences. The success stories existed, but were kept underground. This systematic silencing prevented the obvious question. If bacterial inoculation worked so well for these farmers, why wasn't everyone using it? The industry ensured this question was never asked widely. Number 8, the modern soil, science that rediscovered bacteria. Contemporary soil science is rediscovering what was known and suppressed 70 years ago. Modern researchers are documenting that biological nitrogen fixation works exactly as World War II studies showed. Recent studies using DNA sequencing identify nitrogen-fixing bacteria in healthy, productive soil. Azotobacter and related species are abundant. Their populations correlate directly with soil fertility and crop productivity. Field trials in organic agriculture show bacterial inoculants increasing yields substantially. The effects match historical data. Crops growing vigorously without chemical fertilizers. The biology hasn't changed. The suppressed knowledge is being validated again. But modern researchers rarely acknowledge the historical precedent. They present biological nitrogen fixation as recent discovery. Credit contemporary scientists. Never mention that this was known and deliberately suppressed decades ago. The papers carefully avoid challenging chemical fertilizer necessity. Present bacterial inoculants as supplements rather than replacements suggest they enhance chemical fertilizers rather than eliminate them. The framing protects industry interests. Research funding still comes largely from agricultural corporations. Modern scientists face the same pressures as their predecessors, challenge industry interests, lose funding. The suppression continues through economic control, but cracks are appearing. Independent researchers funded by organic agriculture organizations are publishing stronger findings, showing bacterial inoculants can completely replace synthetic fertilizers. The evidence is accumulating despite industry opposition. Social media allows farmers to share results directly, bypass agricultural establishment gatekeepers. Success stories spread despite attempts at suppression. The knowledge is harder to contain in the information age. Soil health movement is embracing biological approaches. Farmers are experimenting with bacterial inoculants independently. Commercial products are reappearing from small companies outside industry control. The knowledge suppressed for 70 years is resurrecting, not through academic institutions or agricultural establishment, through grassroots farmer networks rediscovering what worked. Number 9, the regulatory capture that prevents access. Modern regulatory systems prevent access to azotobacter and nitrogen-fixing bacterial inoculants through rules designed to protect chemical fertilizer industry rather than farmers or soil health. Bacterial inoculants are regulated as agricultural inputs similar to fertilizers. This requires extensive testing, registration, and approval processes. The costs run hundreds of thousands to millions of dollars per product. Small producers cannot afford these regulatory costs. Only large agricultural corporations have resources to navigate approval processes. This creates barrier favoring established chemical companies over biological alternatives. The testing requirements are designed for chemical products. Bacteria are living organisms. Requiring the same testing protocols as chemicals doesn't make scientific sense, but it creates expensive obstacles to bacterial product approval. Even when bacterial products gain approval, labeling restrictions prevent manufacturers from making claims about fertilizer replacement. Can only advertise as supplements or soil conditioners. The true value proposition cannot be communicated legally. Farmers wanting to produce bacterial inoculants for their own use face prohibitions. Regulations classify bacterial cultures as regulated agricultural inputs. Home production is illegal without proper licensing. Knowledge that should be freely available is criminalized. Universities cannot distribute cultures to farmers. Institutional rules and liability concerns prevent sharing bacterial strains. Research materials must remain in laboratories. Practical application is blocked. Seed coating with bacterial inoculants faces additional restrictions. Could be the simplest distribution method. Seeds arrive pre-inoculated, but requires separate approvals and testing. The obvious solution is made deliberately difficult. These regulations aren't about safety. Azotobacter is naturally occurring and harmless. The rules exist to protect chemical fertilizer market from biological competition. Regulatory capture by industry interests. Breaking these barriers requires political action. But agricultural policy is influenced heavily by fertilizer industry lobbying. The regulatory system serves those who fund it. Farmers and soil health are not priorities. This wasn't experimental agriculture. This was survival farming tested under conditions where food production failures meant mass starvation. No chemical fertilizers available, no industrial inputs possible, just biology that worked or populations died. Soil fertility doesn't require factories, doesn't require mining, doesn't require chemicals. Bacteria provide everything when conditions allow them to thrive. Soviet survivors knew this. Their research documented it thoroughly. For thousands of years before industrial agriculture, soil fed itself through biological processes. Modern industry made us forget, because forgetting generates hundreds of billions. Every biological solution buried is a chemical product sold. Every natural fertility process suppressed is a fertilizer purchase required. But soil remembers. Azotobacter still exists, still fixes nitrogen, still builds fertility, still makes chemical fertilizers obsolete. The biology hasn't changed despite 70 years of suppression. I'll see you in the next video.